My Thank you, Diane. It is my absolute pleasure to introduce Dr. Kimberly O'Brien, who's a professor in the Division of Nutritional Sciences at Cornell University. Kimberly did her BS in biology at University of New Hampshire, and then went on to do a PhD in nutrition at University of Connecticut Stores, followed by postdocs at both NIH and Baylor, where she developed exceptional expertise in mass spectrometry, studying mineral and vitamin metabolism. She focused on both vitamin D metabolism and iron metabolism. She started her career as an assistant professor at Johns Hopkins in 1995, and then she came to Cornell in 2005, right at the time where I started my PhD, which is where I got to know Kimberly. And she has done some fantastic studies on hepcidin regulation of iron absorption in humans. And these studies have really highlighted mechanism of how hepcidin regulates iron availability across the maternal-fetal interface. And today she's going to tell us about this work, determinants of maternal-fetal iron partitioning, new approaches, and unresolved questions. And we're really excited, Kim. This is something that I have I've heard bits and pieces about over the years, and I still, every single time I learn new things. So I'm really excited to hear more about your work. Thank you so much, Kimberly. And for those of you with questions, please feel free to populate the chat, and I'll try to address those or raise your hand if it's urgent. Thank you.
Great. Thank you so much, Anna, for that kind introduction and for the invitation to present in this seminar series. It's exciting. So today I just want to tell you a little bit about some of the things that we've done in maternal-fetal iron partitioning, and I'm going to digress a little bit to one other aspect that I want to talk about today and get your thoughts on if you don't mind. So here we go. So I don't have to tell any of you about iron, but I'm still going to throw out some factoids that I find really fascinating. One of which is that although iron is such an abundant element on Earth, it's one of the most common nutrient deficiencies. I think this becomes particularly important during pregnancy, since pregnant women need this to support their health, the function of their placenta, and optimal fetal development. Unfortunately, many women that are otherwise well-nourished in the United States don't enter pregnancy with enough storage iron to support the 1,000 milligrams of iron that's necessary to sustain a typical singleton pregnancy. Prenatal supplements in the United States contain 27 milligrams of iron, yet anemia and iron deficiency remain common in this country. Recently, we just completed a 10-year retrospective medical chart review at the University of Rochester in over 40,000 women that provided us with longitudinal data on iron status across pregnancy, and that highlighted that even when restricting to otherwise healthy women, a quarter of these women had anemia in late gestation. Another recent study in California looked at medical records from almost 4 million pregnant women and found again that anemia was evident in up to 22% of these individuals. And this is concerning, given that maternal anemia has been associated with major maternal and perinatal morbidity. It impacts maternal health and well-being, and it can have detrimental effects on the fetus and newborn. As you know, recently the U.S. Preventive Services Task Force published their recommendations on iron deficiency and iron deficiency anemia during pregnancy, and they concluded that there is not currently enough evidence to say whether or not asymptomatic pregnant teens or pregnant adults should be screened for iron deficiency and iron deficiency anemia, or if they should take regular iron supplements. Unfortunately, it's been acknowledged that the existing data to inform these recommendations really wasn't sufficient to address 4 of the 5 key questions that were being reviewed. There really is a significant gap in knowledge in respect to even very basic aspects of physiology. We don't have robust screening thresholds for iron status biomarkers, and these are often poorly defined and aren't consistent across different agencies. In addition, some very basic aspects of iron metabolism during pregnancy haven't been well characterized, and much of what we know is based really on early studies, or has been based on early studies. One of the key limitations in this field in general, I believe, is that when you're setting nutrient recommendations, you're looking at the level of intake that's necessary to prevent deficiency. Really, this isn't optimal. We need to look at the level of nutrient that's necessary to support maternal health. And during pregnancy, we also need to look at the level of maternal intake necessary to sustain the placenta so that it functions optimally and to ensure that the newborn contains enough nutrient at birth to get it through its early growth period. And many studies don't consider anything other than the mother. They don't think about the placenta or the neonate in the fetal acquisition. Again, you all are well versed in this schematic, but I just want to talk about a couple things with respect to pregnancy. We don't need very much absorbed iron to maintain homeostasis in a non-pregnant adult, only 1 to 2 milligrams per day, because the vast majority of iron that we utilize to sustain requirements comes from the catabolism of red cells. And to maintain this, it's unique because we can't physiologically excrete iron. We need to be able to sense utilization, recycling, and storage, and then modify absorption to accommodate this system, which involves then the use of erythropoietin, erythrophorone, and hepcidin. As you know, hepcidin binds to ferroportin and can block iron release from stores or iron export across the enterocyte. During pregnancy, up to 8 milligrams of iron per day is shunted to the fetus during late gestation. This reflects almost half of the iron that is released from red cell catabolism. This can be a significant drain, particularly in a pregnant woman that's reliant more on iron to expand plasma volume and support placental function. We haven't had enough studies or enough approaches that allowed us to look at utilization, recycling, and absorption dynamically in pregnancy with safe tracers. And that's something that I've been interested in doing for a long time now, is using stable iron isotopes to better approach these dynamic measures of iron utilization across gestation and to address issues of maternal, fetal, placental iron partitioning. And stable isotopes are naturally occurring, non-radioactive. Iron exists in 4 different atomic weights. 3 of them are found under natural abundances under 6%, and we can give them multiple tracers orally, give tracers orally and intravenously, and we can begin to evaluate all of these different aspects of iron utilization across gestation safely. So for many years, we've been working with pregnant populations, screening a number of iron status biomarkers, measuring all of these hormones as they've been identified, isolating, collecting the placentas, isolating protein and RNA from these placentas, and building an archive that we can really utilize to explore a lot of the basic aspects of how these parameters are being regulated across gestation. We tend to focus, we're criticized often because we focus on higher risk obstetric populations, first because that's what we can get the funding to examine, but second because these are the populations that I think are most important to really start looking at, because we're going to be able to see what happens when the system is strained and how are these parameters regulated in response to that iron deficiency or iron deficiency anemia. So a lot of our work has been undertaken in pregnant teens and in women carrying multiple fetuses, twins, triplets, or quadruplets. And I'll share some of those findings with you today. And again, many of our studies administer these stable isotopes so we can get these dynamic measures. You can see on the little schematic above, we have over 300 pregnant women where we've done these studies. And because we study women carrying multiples, we have over 500 newborns. We work with the obstetric staff at the University of Rochester, who's phenomenal, and we have been able to collect in hiring health project coordinators that are willing to work 24 hours a day and carry pagers so they can go in and collect the placentas. We get about 80% of our placentas from these populations. So I want to just share some of the interesting findings, I think, from these types of studies. So using a stable oral isotope, we can give inorganic iron as ferrous sulfate. We dose women orally, wait 2 weeks, take a blood sample. 80% of that iron is incorporated into red cells, and we can use this measure as a measure of iron absorption. So using this approach, we have looked to see the degree to which pregnant women can upregulate iron absorption as they deplete their iron stores. And while you're able to upregulate iron absorption, because we have this largely closed system, we're pretty conservative about how we do that. And pregnant women only upregulate about 3% for every 10 microgram per liter decrease in serum ferritin, which reflects about 80 milligrams of storage iron. We were able to publish the first data relating hepcidin to iron absorption in non-pregnant and pregnant individuals. When hepcidin was discovered, we had hoped that it would explain a larger fraction of the variability in iron absorption, but it only captured 20 to 30% of the variability, which is comparable to serum ferritin. When looking at all of the biomarkers that we have screened right now, the most predictive biomarker of iron deficiency and iron deficiency anemia in our studies is the ratio of hepcidin to erythropoietin. We believe this is because this is reflecting both iron deficiency and hypoxia. One of the important things in iron that is almost never, except for, some people, that's all they do. But if you look at nutrition studies and you look at setting iron requirements, people think of iron only as inorganic iron, although we all know we ingest iron as inorganic iron and heme iron. And so using stable isotopes, we intrinsically labeled a piglet. I did not realize that piglets at birth get 150 milligrams of IV iron to support their early growth. So we replaced that iron with stable iron. We then took the muscle and the red cells, we made chili and we fed it to pregnant and non-pregnant women. And we were able to look to see how heme iron was absorbed. And using this approach, we then measured labeled iron derived from heme intake and found that heme iron absorption, as has been shown many times, was significantly higher than non-heme iron absorption. We found it was not associated with maternal iron status, and we also found that it was not associated with maternal hepcidin concentrations. I believe that iron can get catabolized in the enterocyte into inorganic iron and exported as inorganic iron through ferroportin. If all of the absorbed heme iron was getting catabolized, we should see a significant association between heme iron absorption and hepcidin, and we do not. And to me, I believe this indicates that much of this heme iron is getting exported, not as inorganic iron. I'll come back to this in a moment. So there are challenges when looking at nutrient absorption during pregnancy. When you absorb a nutrient during pregnancy and you're looking at it only in a maternal pool, you're not truly capturing maternal absorption. You're just measuring maternal utilization of that nutrient. And so in our studies, what we wanted to do is dose and look at maternal utilization by measuring the iron in red cells. But we also wanted to look at maternal absorption by measuring the amount of absorbed tracer that was retained within the placenta or present in the newborn at birth, to get a better measure of actual maternal iron absorption during pregnancy. So we utilized this approach, and we found that if you quantify the amount of tracer in the placenta in the newborn at birth, that we found that if you don't account for those losses, you underestimate absorption by 17%. And that degree of underestimation isn't fixed across individuals. It varies according to maternal iron status. Women that have lower iron stores tend to send more of their recently absorbed iron to their fetus. So the degree of underestimation is not fixed across individuals. Just kind of as an aside, one of the estimations that goes into the dietary requirements for iron is how much iron is transferred and retained within the placenta. The early DRIs used a value that was obtained from very early studies in a very diverse and unusual obstetric population, and that study concluded that the iron content of the placenta was 90 milligrams. In our studies, the iron content of the placenta was only 40 milligrams. Again, you might say that's because we work in higher risk populations, but we also reviewed all of the published literature we could find on this using much more robust techniques than were used to define the earlier value. And it looks like the placenta contains about 40 milligrams of iron at birth. So even basic questions in maternal iron physiology really had not really been a lot of data. So using this enriched heme iron tracer, we also wanted to see how much of the heme tracer was utilized by the mother, but how much of that heme iron tracer was found in the newborn at birth. When we measured heme in the newborn, the heme derived tracer in the newborn at birth, we also found that a greater fraction of maternally absorbed heme iron was found in the newborn at birth. Again, this to me indicates that not all of that heme was catabolized and exported as inorganic iron. So human placental transfer is really unique and distinct from the enterocyte, and I think it's important to highlight some of the unique aspects of placental iron trafficking. First, obviously, the fetus is dependent on placental iron transfer. This transfer across the placenta, across the syncytial trophoblast, is transcellular and unidirectional. Through most of gestation, the syncytial trophoblast is patent, so there are no paracellular junctions, so this is all transcellular. Unlike the enterocyte, which has the luxury of letting iron in, storing it as ferritin, and then getting sloughed off so that humans can eliminate non-necessary or non-needed iron, the placenta either needs to restrict iron from entering or sequester it over the 9 months of gestation if the fetus doesn't need this. Unlike the enterocyte that's exposed to one hormonal environment, the placenta is exposed to the maternal and the fetal hormonal environment, and it makes all three of these regulatory hormones itself. Another unique aspect of placenta physiology is that the placenta has very high energy demands, higher oxygen demands on a per kilogram basis than either the mother or the fetus. So it requires a pool of iron to support its own function across the course of gestation, which automatically sets up some type of competition if iron becomes limiting. Another aspect that's important to consider is if you're looking at flux across this organ, the tempo of nutrient uptake by the placenta is not static. It changes markedly across gestation. This is a figure that we compiled using early chemical composition data from Elsie Whittleson. She chemically analyzed a lot of human fetuses at variable stages of delivery and then mapped their total iron content onto fetal weight. And the first dotted line is showing you that at 28 weeks of gestation, a human fetus weighs about 1 kilogram and contains 90 milligrams of iron. Over the next 3 months, that fetus has grown to over 3 kilograms, so it's tripled its size and tripled its total iron content. So the tempo at which iron is delivered across the placenta is very highly variable across the course of gestation and peaks during late gestation when most of the iron is accumulated by the human fetus. So in addition to the tempo being highly variable, there are also differences in where that iron is getting presented to the syncytial trophoblast. So early in gestation, up until about 12 weeks of gestation, the fetus is obtaining its iron through histiotropic nutrition. The iron is being presented or released from uterine glands. The maternal spiral arteries are largely occluded by extravillous trophoblast, so maternal blood isn't bathing the syncytial trophoblast. It's not until about 12 weeks of gestation that the spiral arteries become patent, release fully their maternal blood into the intravillous space where it can bathe the syncytial trophoblast. And then at this time, all nutrients that are taken up and delivered to the fetus are delivered through hemotrophic nutrition. So this is another parameter to think about when evaluating issues relevant to nutrient trafficking across the placenta and doing so across the course of gestation. It's another dynamic process that's changing. One of the things that my group has also been interested in thinking about is where is that iron coming from? So diferric transferrin in maternal blood can reflect iron that was obtained from maternal recent diet, or it can reflect iron that was released from maternal red cell catabolism. And so we were interested in kind of exploring those differences, and we did that in a study that we pre-labeled women. So early in gestation, at week 14 of gestation, we administered stable oral iron isotope. That iron is going to get absorbed into red blood cells. We then obtained, in that allows us early in gestation to get a measure of iron absorption. But what we also did is we followed labeled maternal red cells over the course of gestation so that we could get a measure of red cell catabolism in these individuals. In the same woman, then late in gestation, we gave them a second stable isotope of iron as iron 58, and we can measure that 2 weeks later in the red blood cell. We administer this late in gestation where we don't believe that these red cells are going to be catabolized, so that we can then compare the 57 iron and the 58 iron dynamics in a few different ways. So some of the first things that we found using this approach is first we were able to get longitudinal data on iron absorption. And as one would predict, you increase iron absorption as gestation progresses because you're depleting your iron stores. And this increased more than twofold in these individuals across the course of gestation. Again, as one would expect, the increases were most robust in women that had no serum or less serum ferritin and lower hepcidin concentrations. By following the clearance of the labeled red cells over gestation, we found the labeled red cell turnover averaged 95 days, which is a bit shorter than has been reported in non-pregnant individuals. But what we noted is that the loss of red cell enrichment was more robust in women that had low iron status, as measured by total body iron, or as indicated by a higher absorption early in pregnancy, and that occurred in women with lower iron stores. One, again, of the unique aspects of this approach is that we can label women, and then we can collect cord blood at birth, and we can then measure the amount of tracer that was transferred across the placenta and retained within fetal red blood cells. So doing this approach, we could look at slopes of loss in red cell enrichment, and then we could look at the amount of isotope recovered in the newborn, and we found that individuals that had a greater loss of enrichment over gestation had a higher enrichment of the tracer in their fetus. We then wanted to see if we could try and estimate the net quantity of iron that was released from red cell catabolism and that was delivered to the fetus. And using calculations, it looks like women were transferring on average about a third of the iron that was getting released from red cell catabolism was getting delivered to their developing fetus. And again, this was associated with maternal iron status. And I suggest in showing that maternal red cell pool serves as a significant source of iron for the fetus, particularly in women that have depleted body iron pools. With our placental archive, we were interested in trying to identify determinants of the expression of transferrin receptor and ferroportin, and looking at how the protein abundance of these proteins was influenced by maternal, neonatal, or placental determinants. We and many other groups have found that transferrin receptor expression increases in women that have iron deficiency. So in iron deficiency, more transferrin receptor is expressed in the maternal villus membrane, allowing the placenta to better capture dipheric iron. We found that neonatal iron status and hepcidin weren't associated with transferrin receptor 1 protein abundance, so it's responsive to maternal status. Using our archive and then using an Amgen antibody for ferroportin, we screened 263 placenta samples from newborns, and we found that newborns that had poor iron status or indications of increased erythropoietic activity had greater placental protein abundance, and that of all of the parameters that we measured, fetal hepcidin explained the largest proportion of the variation in placental ferroportin abundance. And this is an area that has a lot of controversy in the literature with respect to what is the basement membrane ferroportin responding to. In our studies, it's highly significantly associated with neonatal iron status and not maternal iron status. One of the other unique parameters that Ella Nemeth has developed is the placental iron deficiency index, where you can look at the ratio between the exporter to the importer and use this to get an index of placental iron deficiency. In our studies using this metric, we find that maternal anemia and increased erythropoietic activity in the mom is associated with a lower placental iron deficiency index. Many studies looking at this aren't measuring neonatal iron status. If we look at our neonatal indicators in our populations that have a higher prevalence of anemia than many, we do find that neonatal anemia and increased erythropoietic activity is associated with higher placental iron deficiency index, more ferroportin than transferrin receptor expression, suggesting that ferroportin is responsive to fetal iron status. It's hard to, there's many challenges in studying iron deficiency in the placenta and many reasons that may explain some of the variable findings that come out of this literature. First of all, if you think about the timing of iron deficiency in a human study, this is highly variable. Women may enter pregnancy with anemia or iron deficiency, or they may never develop it, or they may develop it very late in gestation. And there are different gradations of iron deficiency severity or anemia severity. When sampling the placenta, there are not like one gold standard protocol that groups are using. Some people may be sampling by cord insertion sites. Some are sampling more at the periphery of the disc. And this may also add variability into the metrics that we measure in this organ. When we collect syncytiotrophoblasts or tissue from the placenta, we're not just getting syncytiotrophoblasts, but the placenta contains many other cell types that can introduce variability into these measures. In a human study, we only get this tissue at term. No one's donating placental tissue to us early in gestation. So this only allows us to look at this dynamics and these metrics at delivery. In a human study, we have women or individuals with anemia often have multiple other nutrient deficiencies that can be influencing the results that we get and making it harder to make conclusions from our data sets, whereas in an animal study, you can only cause a deficiency of just one nutrient and make sure that other nutrients status is fine. Mode of delivery in a human study is not controllable. We don't tell someone how they're going to deliver. In an animal study, most of the animals are taken by C-section. Another thing that often isn't considered is the sex of the placenta. Many studies now have shown that proteomic and transcriptomic changes are different in response to nutrient deficiency differ between male and female placentas. And this is something that often also may not be fully considered in interpretation of data. So a lot of additional challenges in trying to interpret and making it hard to do human studies where we can really address all of these parameters. So one of the questions that we've really been interested in looking at is who's controlling this dynamic? To what degree does maternal status, neonatal status at birth, and placental parameters influence partitioning of iron between these three individuals? If you just have a singleton birth, we only have one fetal placental unit and one maternal hormonal environment. And this is why I think looking at a human multiple birth model allows us to kind of answer a few additional questions that we wouldn't be able to look at in a singleton delivery. And one of the advantages to focusing on multiples. So we have recruited today, we did a study in 83 women carrying twins, triplets, and quadruplets. As you would predict, there's a lot of anemia in these women. 45% were anemic. At this time, there are no special iron supplementation recommendations in women carrying multiples. They were the same as women carrying singletons. And at delivery, these 83 women gave birth to 183 newborns. We try and get placental tissue and cord blood from everyone. Sometimes there are multiple other issues going on at birth that preclude us from getting some of those samples, but we obtained cord blood from 144 babies at birth, placental tissue. 12% of them were anemic at birth. 70% were born by preterm and 74% C-section. So in this population, what we wanted to do is we wanted to look at intraclass correlation coefficients and evaluate the degree to which iron status parameters what was the variability, the variance in utero between womb mates compared to the variability that we see between unrelated newborns. And this approach is used in the animal industry a lot when you're trying to identify factors that can maximize birth weight. Values close to 0 are suggestive of more variability within a uterine cavity between siblings than between unrelated neonates. Values close to 1 indicate that there's more variability between unrelated newborns than between siblings. When we used this approach in our multiple birth cohort, we found that cord hepcidin concentrations were more variable between siblings than between unrelated neonates. And then when trying to identify what were the significant determinants of iron status biomarkers in hemoglobin, and we focused on hemoglobin in a newborn because that is the largest iron pool in a newborn. The fetus prioritizes iron for hemoglobin production so that it can extract oxygen from the maternal blood. And looking at hemoglobin, cord hepcidin explained the greatest fraction of the variability in hemoglobin between siblings. I mentioned this already, but placental ferroportin in this multiple birth cohort was significantly associated with neonatal hepcidin. It was not associated with maternal hepcidin. It was not associated with placental iron content or neonatal or maternal ERFI. So with all of the data that we have so far, with all of the evolving regulators of iron homeostasis, we still are capturing only about 30% of the variability in iron status or iron absorption. And there are many, many questions that remain on maternal-fetal iron partitioning. And I just wanted to digress a little bit here and tell you a little bit about another study we're doing right now that's addressing genetic determinants of iron absorption, and then loop back to why this has relevance for pregnancy. So because we can't physiologically excrete iron, if we don't tightly regulate absorption in response to our iron demands, we can develop iron overload. The risk of elevated iron status varies by ancestry. So hereditary hemochromatosis is due to a mutation in the HFE gene. This is more common in Northern Europeans, and this mutation is nearly absent in Asians. About 20 years ago, the hemochromatosis and iron overload screening study recruited 100,000 individuals across a wide age range. The majority of the individuals in this study were over the age of 40. Looking at excessive iron stores, people don't tend to worry about this until individuals are mid-age or later. So this was a relatively older cohort. One might hypothesize that the risk of elevated iron stores would be most evident in Northern Europeans because they carry this HFE mutation more commonly. But this EARS study found, surprisingly, that the highest risk of elevated serum ferritin or transferrin saturation was observed in individuals that self-identified as Asian. It's also known that individuals of East Asian descent have a greater risk of chronic diseases that are associated with iron overload, such as type 2 diabetes and others, and that these diseases manifest at a lower body mass index. So to date, there's been a handful of GWAS studies looking at iron status biomarkers in European populations. The last one was published in 2022 in over 200,000 Europeans. There are far fewer studies that have evaluated GWAS of iron status in non-European populations. This is a very Eurocentric data. Only one study in 3,000 East Asians. And with the genome-wide significant loci that have been identified, there's 119 that are significantly associated with iron status biomarkers, but collectively they capture about 2 to 6% of the variance in these iron status biomarkers. Many of the genes identified map onto these pathways of iron utilization, such as utilization, recycling, storage, blood loss, but many genes aren't clearly identified as to what their relevance is with respect to systemic iron homeostasis. So we obtained funding from NIDDK about 5 years ago to address the question of iron genes in East Asians and Northern Europeans is what we call the study, or iron genes. And the goal of this study was to see if we can identify genes that are associated with a functional measure of iron utilization instead of focusing on static biomarkers. And we focused on East Asians and Northern Europeans because East Asians have the greatest risk of iron overload. We finished the clinical studies. We screened over 2,200 people to get our 504 person cohort. We genetically confirmed ancestry in this group at entry. They ingested a stable iron isotope. We took a blood sample 2 weeks later. We measured a full panel of regulatory hormones, iron status biomarkers, inflammation markers, got diet intake information from these individuals, and then isolated DNA, confirmed ancestry. And I wanted to tell you a little bit about our data. We haven't published it yet. Our first 3 papers all got accepted in the same volume of AJCN and are probably going to come out next month with an editorial, and we're really excited about that. And we're also excited about some of what we have found in this group and what we hope to do next and how it may be relevant to pregnant women. So unlike the AIR study, the majority of our population was under the age of 30. We had hoped to have more older individuals, but we did this study during COVID. We're recruiting on a college campus, and we ended up with a disproportionately younger population, which we thought might have been detrimental if our goal was identifying iron overload. Our first paper that we're putting out on this population deals with iron status biomarkers. And this paper is largely confirming what we knew from the AIR study. We found that East Asian individuals, East Asian men, had significantly higher total body iron and elevated serum ferritin. We only saw the difference in the males, not the females. And what we were surprised to see, because our group on average was 27 years of age, 17% of these men had excess iron stores. They had serum ferritin concentrations over 300. This is not due to inflammation as measured with CRP or IL-6. So that just confirms what we already knew. We then measured erythropoietin, EPO, and hepcidin in this cohort. We found that East Asian males had significantly higher hepcidin, and both men and women had a significantly lower hepcidin to serum ferritin ratio. So it looked like the ability to upregulate hepcidin in response to growing iron stores was blunted among our East Asian individuals. We also saw significant differences in ERFI between these 2 ancestry groups. Then when we measured iron absorption, we found that if we looked at non-heme iron absorption corrected to a fixed amount of serum ferritin, this was significantly higher in both East Asian men and East Asian women. And then when we focused on the subset of our East Asian male population that had excess iron stores, you would expect that group to have pretty much dampened down iron absorption, but only one of those 21 individuals had low iron absorption. The rest were continuing to absorb a significant amount of iron in spite of their excess body iron. And when we started this grant, what we believed was occurring is that East Asians may have an evolutionary advantage as they evolved in a low iron plant-based environment where it would have been a benefit to be more efficient at absorbing iron from a given amount of iron. But now that we don't have a limited amount of iron in our food supply, this may predispose some individuals to excess iron accumulation. And these young individual, otherwise healthy males that would not ever get screened likely for iron stores were already having a significant accumulation of iron in their bodies. And this is just a figure from the East Asian male cohort. In all other groups in this study, we see the significant linear association between ferritin and absorption, which we always see in our studies, but we did not see a linear association in our East Asian male cohort. In this cohort, there was an inflection point at 162 micrograms per liter of serum ferritin. Below that, there was a linear association. Above that, there was not. And the red dots on this figure are showing the individuals that had elevated iron stores. And you can see that they have not downregulated their iron absorption in response to their accumulating stores. We're in the process now of trying to identify some of the genes that may be associated with these findings using candidate association analyses. I'm doing this study with one of my former graduate students. I was on his committee when he was in graduate school. He's now a professor of genetics, an associate professor at the University of Georgia, and it's a real pleasure to be able to work with him on this study. His name is Kai Zhang, and Kai Zhang Ye is helping us with this. So what we have done is using those 119 significant loci that have been identified from the GWAS studies in Europeans, we have restricted it to the ones that have minor allele frequencies over 1 and where we have good imputation quality, etc. And then we have mapped on from these genes the ones that are significantly associated with the iron status traits and with iron absorption, and that's shown in these Venn diagrams. And there's a few important preliminary points that I want to highlight. The first is we found no overlap in significant genes between these ancestry groups. And again, I think this emphasizes the need to really have more diversity in our GWAS studies. The other finding is we found significant associations between HFE genotype and iron absorption or serum ferritin, as one would predict. And then we found 7 genes that were significantly associated with iron absorption in the Northern Europeans and 5 associated with iron absorption in the East Asians. And we're in the process now of more fully exploring these findings with Kai Zhang and with Alexa Barad, who is the graduate student who has been working on this project and who is first author on all of the 3 papers that I just showed you. She has since graduated. She's now doing a postdoc at Stanford. So I want to loop back to what we were talking about initially. I don't do a lot of work or as much work in non-pregnant individuals. Pregnant individuals are far more fascinating to me, but this I think has a lot of relevance to iron supplementation during pregnancy. We focus really on looking at distributions and coming up with a measure of iron intake that would be beneficial for the entire population. For iron, I think there's some unique challenges here because of iron physiology. If we cannot excrete it, if it's universally supplemented, and there are some groups that may be predisposed to accumulating more iron than others, this may have some unwanted detrimental effects that it's difficult to really account for and may be where we're going with precision nutrition approaches. I want to just show the schematic from our data at the University of Rochester in the 40,000 women where we had longitudinal measures of hemoglobin over gestation. That's the figure here on the right. And we grouped women according to their self-identified group of ancestry as Asian, Black, other, or white. What we found, like many studies find, is that Black individuals had the highest risk of anemia, lowest hemoglobin at entry, and highest risk of anemia at delivery. We found in our Asian cohort that our Asian individuals had a different tempo of hemoglobin change across gestation with earlier nadir and a quicker rebound and significantly higher hemoglobin concentrations at term. Other studies have also found this from much larger cohorts. So I think it's just an issue that needs to be further considered when looking at iron homeostasis and dynamics during pregnancy. So just to conclude some of the key issues, pregnancy is such a unique life stage. The requirements and nutrient dynamics at this stage of life need to consider the impact of nutrient exposure, not just on the mother, but also on the placenta and on fetal outcomes. We need to be better about considering dietary iron exposure during pregnancy or any other life stage as not just exposure to inorganic iron, but to think more comprehensively about the types of iron that we're consuming and ingesting. We need more data to better characterize the impact of many other factors that are going to be influencing iron partitioning and utilization between the mom, placenta, and fetus, such as fetal sex, hormonal environment, maternal age, obesity, etc. And as the literature evolves, I think it's important to be thinking about these precision nutrition approaches and to be considering genetic factors that are influencing nutrient uptake and utilization across pregnancy. And I want to acknowledge the many, many individuals, many doctoral students had generated the data that I've shared with you today. I mentioned Alexa, who worked on the Iron Genes Project, and Kai Zhang Ye, who's assisting us at the University of Georgia. All our erythropoietin data was from a multiple PI grant that I had with Ella Nemeth and the many, many clinicians at the University of Rochester. Key among them is Eva Pressman, who's the head of obstetrics that I've worked with for several decades now. And also, I also want to acknowledge our undergrads. Cornell, our division has 700 undergrads. Many of them are pre-med, and they are absolutely phenomenal in helping us undertake these studies and recruit and process. And they also translated our flyers into Korean and Chinese. They're just a very talented group. So I'm going to stop there and thank you very much for your attention.
Thank you, Kimberly. That was fantastic. Super comprehensive and interesting. Questions for Kimberly? Oh, okay. That's clapping. Well, please go ahead, Elena.
Hi. So thank you so much, Kimberly. That was, as always, wonderful tour de force. My
Yeah.
question is about the East Asian males and whether the data demonstrates any confounding by um, whether ferritin release from intracellular to the serum where it's measured is altered, or whether the serum, a ferritin expression, the regulation of ferritin expression is more robustly enhanced in response to iron, enabling greater tolerability of higher iron levels. Um, I'll stop there.
Yeah. How would you propose that we explore that possibility with kind of biomarkers that we have access to? One of the things that we have looked at diet and exposures and what are the most significant, if are there dietary factors that explain this? East Asians are generally thought not to have as high a protein intake. They had higher protein intake. We didn't have differences in iron intake, total iron intake between the groups. But can you think of how we might explore that really interesting question with the biomarkers that we have?
I think looking at transcriptional regulation would be one way. The secretion question, that's yet another, and that would be in culture, of course, to take perhaps, I don't know if hepatocytes are appropriate, or generate a mouse model that then allows you to, I'm not suggesting that these are
Yeah,
simple questions.
yeah,
I'm just curious to what degree that some of those were initiated to
yeah.
think through.
Yeah, I think they're all really interesting questions, and this gets to some of the limitations of what we can do in our human studies and what we can do with the budget that we get to do our human studies. So by the time we budget up to do this study, we didn't have additional resources that we could combine animal models and more mechanistic studies, map them onto what we were doing, and that's a limitation. And then for this particular study, NIH, one of our reviewers told us that we could very easily recruit our 500 people in 4 years. So they cut a whole year off our project. So we just didn't have the resources to look at that. But those are really important things that from what we have, we haven't been able to identify anything that could easily explain some other measure that might be driving the ferritin differences that we see. There's no differences in inflammation. Actually, the Northern Europeans have higher CRP, but if we control for body weight, those differences go away because the Northern Europeans weigh more than our East Asian participants did. So we haven't been able to discover any obvious reason for those differences. We also have menstrual loss questionnaires that we're in the process of exploring in our female participants, but so far I can't answer your question, and we don't have a data to do that, but it's a very important question to
Yeah, no,
pursue.
without a doubt. And I didn't mean any criticism in even one iota of thought about any kind of criticism about the excellent effort that's already been made. I come from considering renal failure patients and the differences, not just in the practice of administering IV iron in renal failure in Asia versus in the US, but appears to be perhaps to your point, a physiologic difference that requires different amounts of parenteral iron administration to reach similar anemia management goals. And so there's an overlap there with other areas to investigate where your expertise might be extremely useful in understanding the genetic underpinnings that you're presenting.
I never thought of that. I'd like to talk to you more about that. That's
I
really
look forward to that.
Yeah.
Go ahead.
I had a quick question. Do you hear me?
Yes,
Yes.
I can hear you.
We have a model from a long, long time ago that of severe iron deficiency, dietary iron deficiency in mice, and we look at kidney development. And one of the things is that at some level of iron deficiency, there's enough tissue sort of dysplasia that we think it's really inflammatory, and hepcidin levels are gigantic in that setting, and probably impact placental iron transfer. Is there any evidence in human data that iron deficiency in gestation and the newborn is actually an inflammatory condition at high hepcidin levels related to that, or that's not seen in humans?
We have seen that our iron deficient neonates have low hepcidin concentrations, and it looks like the newborn is appropriately regulating hepcidin in response to its iron that it's receiving. So we haven't seen that. We haven't seen that it looks like there's inflammation in the newborns.
So this model is with such severe iron deficiency that there's a lot of tissue destruction, and maybe in that setting, macrophages, etc., are activated. We see a ton of hepcidin, but that's maybe very, very far beyond anything a human would suffer.
About a quarter of our babies have anemia at birth. And this is another thing that I think we just have this dogma that all babies are going to scavenge everything they need and be born iron replete. Doesn't matter what the maternal iron status is, but in the groups that I'm talking about, up to a quarter of those babies are anemic at birth, even in term babies. So it's not that they're preterm, and they're not going to be screened unless they're low birth weight or have other complications that would necessitate screening them. So I don't think we have enough data on iron stores at birth and on who should or should not be screened to really know even how to interpret iron biomarkers at birth. In our studies, ferritin is inversely associated with hemoglobin and cord blood at birth. So babies with low ferritin have high hemoglobin, babies with low hemoglobin have high ferritin. But ferritin is used often as an index of iron sufficiency in cord blood. So there's just more normative data I think is needed to understand what all of those biomarkers are telling us, because they change very dynamically over the first week of life. You're catabolizing hemoglobin and redistributing all of that iron released into stores, but at birth you have prioritized it into hemoglobin from the fetus. It's complicated. Yep.
Kim, I got a question. It's the assumption that in non-pregnant individuals and healthy males or females, 90% of the body iron comes from recycling and 10% from absorption, right? But you made the comment that a third of the iron that's coming from recycled iron from red blood cells is shunted to the fetus. So to me, when I'm thinking about pregnancy, I'm thinking about non-pregnant individuals that's non-anemic as the control. Right. So how does this equate to, is this a surprising result that a third of the fetal iron is derived from recycled iron? I would have thought even more so.
Yeah. I don't think it's surprising. And it was variable, so a third on average, and we have a population where they're not all anemic, right? So in our multiples, half were anemic. In the other group, it was less than half, maybe a quarter. So we're looking at an average from a very diverse population across the entire spectrum of iron status. And I agree with you that that value could get much higher in an individual that doesn't have stores or may not be taking in dietary iron. That was an average value. I don't, so it doesn't surprise me that the red cell pool is being scavenged in that way, right? That makes sense. We know that that iron is going to be released always over the course of gestation. It's going to be there, whereas there's less certainty that women may be ingesting iron of heme or non-heme origin over the course of their gestation. But these are a relatively small study. It didn't have as many women as we would like. We did this study just from discretionary funds, didn't have a large number of women. It would be interesting to reevaluate that, being more deliberate in the women that we're recruiting to do this study, and so that we can more fully examine the parameters that we think would be most influencing the dependence on that source of iron pool.
But in this case, when you say heme iron, and to me, heme is heme and not heme iron, but for the iron people, let's keep it separate, right? So, so is this really iron that's being transported to the fetus, or is it heme that's being transported to the fetus? And I want to also follow up on your JN 2012 paper where you said, which was very interesting to us, is that heme gets preferentially shunted to different organs, absorbed heme versus iron. So have you followed up on those studies in terms of heme absorption versus iron absorption?
No, again, we just haven't had a grant to do that. So we haven't followed up on that since that study. I would love to follow up on that study. We did, you probably saw, we did do a rat study where and that there were differences in where the heme went from an organ standpoint, and a different prioritization or distribution of the heme, labeled heme. And it may not all be, we're calling it all labeled heme, but that labeled heme probably had some non-labeled non-heme in it, right? We took the muscle and the red cells and mixed it together and fed it to someone. So it was disproportionately heme, but not entirely heme. But it needs to be followed up on. And it would be best to follow it up on with a joint human animal model study, so that we could inform the animal studies to get, to support what we're seeing in the human studies and be able to figure out exactly what's going on.
Thank you.
I'll take the last question if I can. That was a great talk, Kimberly. And you mentioned that iron deficiency often co-occurs with other nutrient deficiencies. So I have to ask if you've looked at how folate deficiency overlays with this, especially since it regulates erythropoiesis and potentially iron utilization from red blood cells.
Yes, your mentor would be so proud of you. Yeah. So we do measure folate and B12. They're not folate deficiency. They're not folate deficient in general. This group, they're eating a lot of processed foods. They have deficiencies in vitamin D, and there's very little B12 deficiency. So I don't think that folate deficiency is one that is influencing the dynamics that we're seeing in this particular population. But in other populations, it may further exacerbate this situation, but it doesn't in the groups that we've looked at. It doesn't seem to. We don't really see even much folate insufficiency.
Okay. I thought of it as you were talking about, and I loved that idea of the tempo of nutrient uptake across gestation, and how I imagined how folate might fit onto that, and uptake of folate across the maternal-fetal interface relative to iron across.
Yeah. And maybe it changes. Maybe there's different, for all of these things, maybe there's different gestational windows where you need to be more aware of the dynamics between interrelationships between these things.
Correct. Super interesting. Well, thank you so much, Kimberly. That was an extraordinary talk. Really appreciate you giving us your time here for the CIHD. And thank you to so much to everyone attending and to your thoughtful questions as well.
Thank you very much.
Yes.
Everybody
See you
take
all
care. Nice to see
month.
all.
Yes, thank you so much.
Bye-bye.
See you guys. See you. Bye-bye.